Right, this lecture is going to cover the chromosomal basis of inheritance, which is looking at genetics at a kind of a larger scale when we're looking at whole chromosomes and what's going on with those chromosomes in genetics. So it's following on from what we've learned, but we're going to learn about a few nuances and interesting situations of inheritance. So we're going to just have a quick recap on gametes and Mendel's law of independent assortment, which is really important to know, especially for this lecture. We'll cover something called wild type versus mutants, and we're going to follow a species called Drosophila, which is fruit fly. And then we'll talk a little bit about sex-linked genes and then linkage. And this lecture originally covered alteration of chromosome number and structure, so when we get things like Down syndrome and stuff like that. But I'm not going to test you on that. However, the slides are in your original lecture, which I recommend you look at because it is super interesting. And then mutations and how mutations happen. We're going to cover that when we do protein synthesis. So throughout this, I'm going to ask you questions like I normally do in class, and I really recommend that you stop and pause the video and try and answer these questions, because the more recap you do, then the more you can identify what you know, and more importantly, what you don't know. So have a go at answering these questions. We've got an individual, let's say it's a bird, and it's got two genes that we're going to follow, gene A and gene B, and these genes are on different chromosomes. So what you already know about homologous pairs of chromosomes and how they differ and how they're similar, etc., try and draw the chromosomes for this individual. It's got two copies of every gene, it's got a dominant and a recessive copy of A, and it's got two dominant versions of B. And then draw the gametes that this individual could make, and remember the FOIL method that we did. You can write the gametes using that. Try and recap on Mendel's law of independent assortment. What is that? And Mendel's law of segregation. And then remember, you've got 23 pairs of chromosomes. Not all of them are homologous. Which ones are not homologous? So pause the video, see if you can do this. This is what you should have for the chromosomes. Remember that every organism that reproduces sexually, and that's what we're talking about in all of these genetics lectures, they have pairs of chromosomes, one from the male and one from the female, that reproduce to make that organism. Okay, and so these are the chromosomes, and it'll have other chromosomes as well, but we're just interested in these genes right now, and we said they were on different chromosomes. All right, so you've got one pair here. This is one homologous pair, and it has the A gene on, and these are each copy of the A gene, one on one copy of the chromosome, one on the other copy of the chromosome, and the same with the B. Right, let's take this through meiosis to look at what the gametes would be. So remember that the X shape that we only see that at S phase, after S phase of interphase. And so let's duplicate those chromosomes. Remember that the duplicated copies remain attached together at the centromere. So these things here, these are the sister chromatids. You've not got any extra information here. The analogy, like I said before, is like copying your biology textbook. Yeah, you might have like 2 copies of every paragraph now, but you haven't actually gained any more knowledge or lost any knowledge. All right, now let's take these through meiosis. So remember the meiosis 1, that is going to separate homologous chromosomes. So this A here is going to be separated from this big A here, and this B from this B here. Like that. And which one goes with which is random. And remember, crossing over would have occurred as well, so these might be different. I've just not drawn it on here. So this is meiosis 1. And then meiosis 2, remember, it's just like mitosis in terms of what happens to the chromosomes. Imagine mitosis on a gamete. We're just going to pull those chromatids apart. So we're going to do that. Okay, and again, it's going to be random every single time, but those are the gametes, the possible gametes here, and we know we might have crossing over going on with other genes, but the important thing is that you've just got one chromosome where the A gene is on and one chromosome with the B gene. So this is Mendel's law of segregation, that these homologous chromosomes, the one from the mother and one from the father, are separated during meiosis, and it's meiosis 1 that's going to separate those homologous chromosomes, where you go from diploid to haploid. So gametes are only going to have one allele for each trait, one copy of every gene, because if they didn't, well, then you would have 2 copies of every gene, and then when the gametes fuse together, you would have 4 copies of every gene, and that's not what a human is, or organisms that reproduce sexually. Okay, now these are the chromosomes that you have that are not homologous. These are the X and the Y chromosomes. The Y chromosome is a lot smaller, and that's going to be important as we go through this lecture, because you'll see a difference in the probability of males and females getting genetic disorders that are on these sex chromosomes, and that's because these chromosomes are different to one another. Okay, so we're going to follow some genes in fruit flies, the poor fruit fly. Fruit flies are fantastic because they only have 4 chromosomes, but that means that they're really simple to study in terms of genetics, and therefore there's been loads and loads and loads of experiments been done on these flies. Some of them really, really cool, like growing like legs on their head and stuff, when we're looking at how development is controlled. And that's going to be in the chapter on regulation of gene expression. All right, so the reason why Drosophila are also studied, they only have 4 pairs of chromosomes, but they have loads and loads of babies. They reproduce every 2 weeks. You can tell the sexes apart, which is really important. So wild type means like the naturally occurring form, because we're going to look at some mutations as well. And so wild type is basically the unmutated form. And this is the female, and you'll see her abdomen is different from the male. The male has this really dark and shorter abdomen here. So that's really important. If we're going to follow genes that are caused by sex chromosomes, well, then we're going to want to follow males and females, and you want to be able to sex them really easily. And so it's called sexual dimorphism, and you see it a lot in birds and things like that, where the male looks different from the female. All right, so this is the Drosophila, and we're going to do some experiments. And a guy called Thomas Hunt Morgan followed these fruit flies and did these genetic experiments. He's kind of like the equivalent to Mendel. All right, so here's a wild type, and it's the most commonly observed phenotype. If you remember, the phenotype is the physical representation of the genotype. Right, so here's a wild type, and you can tell it's a she because her abdomen is larger and not completely black at the end, like the male. So what a mutant is, it's an alternative to the wild type. Something about the fly is different, and when you're doing genetic experiments to follow how traits are inherited, you're often going to want to follow phenotypic traits that are really easy to see. Because you don't want to follow something like metabolism. That's really difficult to study. So what a mutation is a change in the DNA sequence, and that leads to a change in the protein. And that, in the case of this fly, in the traits we're going to follow, is going to lead to a change in the appearance of the fly. Mutations don't always lead to a change of appearance, of course, because your proteins do many, many, many things in your body, but we're going to follow mutations that look different, so they're really easy to tell. So if we were to follow some traits in this fly and these Drosophila, you'll see it's a really kind of a cool looking fly. These are the little things that you get if you leave your fruit out. You get those little flies flying around. This is exactly what they are. So we can follow things like eye colour. Eye colour is a really good one. We can follow things like abdomen size, and then wings. There's lots of mutations that look at the wings, like the shape and the length, whether they're present or not. Some really cool things. So here's just some examples. You don't need to learn these, but we're going to follow some of these. So you can see you can change the eyes, the wings. This one's got tiny little wings. This one's got something going on with its abdomen. Vestigial wings, this is one thing we're going to look at later. Vestigial means tiny little wings that don't really work. And these are all caused by genes. Like cut wings, no one's come along and cut the wings. The fly develops like this. Okay. Right, I have some slight bad news that the way that we write the alleles and, like how we follow alleles, we don't write them the same way that we write other alleles with the capital letter and the lowercase letter. And this is just because this is the way the lab that followed these fruit flies decided to write the alleles, so we'll blame them. But you do need to know this, okay? So I'm gonna tell you about fruit flies when we're looking at wild type mutations, and then we're also going to follow sex-linked traits, and that's written in a different way as well. So make nice detailed notes on how you write these different traits. So in the fruit fly, we're going to use the symbol. We're going to use a letter that represents basically the mutation. And I know that goes against what we've done before, but just learn that this is a special case of writing things. It doesn't really mean anything different. We're just going to write it differently. Okay, so the normal wild type is red eyes. Here's a normal, here's a female with red eyes. There she is. Isn't she beautiful? So she's got red eyes. And the first mutation that was found in eye colour was white eyes. And here's a male. There is his little abdomen there. Okay, they're obviously deep in conversation. And he's got white eyes. And so the mutation for white eyes or eye colour is represented with a W. So eye colour in Drosophila is represented by a W because W stands for white, and white was the first mutation found. Now we want to distinguish a mutant from a wild type. Remember, the wild type is the typical phenotype that we find just naturally. And so if it's got a plus after it in superscript, like a little plus, that means it's the wild type. All right, so this is wild type. So if you see a W plus for eye colour, that means red eyes, wild type. The white eyes is the W, and there is no plus there. Okay, so here the mutant allele is represented by a little W, and the wild type is represented by a little W but with a plus. All right, we'll follow some more traits and practice this more. All right, so what does the W stand for? It stands for the first mutant that was found, which is white eyes. And we put a plus to distinguish the wild type from the mutant. And in this case, white eyes is recessive. And we're going to follow this trait as we go along in these flies. All right, so here is a female. There she is with her white eyes. There she is there. There she is. And she's going to have baby flies. She's going to have some maggots with the male here. And the male has white eyes. Right, so given what you know about Mendelian genetics so far in genetics, I want you to do this cross and figure out what the offspring are going to look like. White is recessive. Red is the normal wild type dominant. Okay, so cross these flies and see what happens. And these are true bred, so this only has the gene causing red eyes and this only has the gene causing white eyes. All right, have a go, see what you come up with. Right, this is the answer, and you probably would have got this. All of the F1 generation, all the offspring have red eyes. So this looks like our typical cross because she's going to give a dominant allele and he's got a recessive allele. And so everyone's going to get a dominant allele because she's wild type. So that's what we expect to happen normally. All right. So they're the parents. Here's the F1 generation. And what I actually want you to do is cross these two together now. We've got a male with red eyes and a female with red eyes. I mean, we predicted that everyone would have red eyes. And I want you to try and do this now. So what you already know predicts the F1 cross. So crossing two F1 together and a male and a female. How many females with white eyes would you expect? And how many males with white eyes would you expect? And I want you to just remember that in sexual reproduction, where we've got two sexes, it's a 50% chance of having a male and having a female. So we would expect the same ratio. So pause the video, try and do the Punnett square of the F1 generation, these guys together, and see what you end up with. Okay, so if you have followed Mendelian genetics and how we've done this in the past, you probably would have got something like this. And so you would have predicted from the heterozygous cross 3-to-1 phenotypic ratio of dominant to recessive. So 1, 2, 3 are going to have the, remember the plus is the wild type. Okay, so red, 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 and 1 white. And we predict 50% are going to be females and 50% are going to be males. So half of the flies with white eyes are going to be females and half of the flies with white eyes are going to be males. So this is what actually happens. You don't ever get any females with white eyes. You predict that you will get them, but when you actually do the experiment, you get males with white eyes in the normal ratio that we would expect, but we never get any females with white eyes. So something else is going on apart from our typical Mendelian genetics, and it's only affecting the males. So what this is an example of is sex-linked genes. So remember, you've got your 23 pairs of chromosomes, but only 22 of them are homologous. The X chromosome is different from the Y chromosome. And different species have different ways of determining sex. They don't always have X's and Y's, and I'll show you that in a couple of slides. But basically, the male sex chromosome is different from the female sex chromosome. Remember that autosomal means you're normal, 1 to 22 pairs of chromosomes. So because the X and the Y chromosomes are different from one another, and you saw that the Y chromosome is a lot smaller than the X chromosome, they have different genes on them. And this makes sense because if you think about it, the only difference between a male and a female is the fact that a female has 2 X chromosomes and the male has 1 X and 1 Y. And so that's the only difference between those two organisms. And so there must be something about the sex chromosomes that's going to be determining the sex, whether you're a male or a female, and therefore they have different genes on them. I'm going to add just another layer of complexity here with writing our alleles. So remember, we're going to stick to the same thing where we've got a little W and we've got a plus or no plus, depending on whether it's wild type or not. So the plus means wild type, and the letter is going to be the first letter of the first mutant that was discovered. So in this case, we're following eye colour, and eyes are normally, the wild type is red, but the first mutation that was discovered was white. And so we're going to represent red eyes as W plus, and white eyes is just going to be W. We're going to write the allele here as a superscript to either an X or a Y, depending on, which chromosome it's on. So here, what this means is this fly has an allele that causes the wild type red eyes, and that gene is found on the X chromosome. So I want you to pause the video again, and I want you to try and figure out the answer to this question. On which chromosome do you think the gene for eye colour is located? Is it on the X chromosome or is it on the Y chromosome? Remember, female is XX, male is XY. And it's only on one of them. I know I was talking about here, it's on, the X or the Y, but this is like, if it was on the Y, this is how we would write it, and if it was on the X, this is how we'd write it. The gene is only either on the X or the Y in this fly. So remember, only males in this cross can have white eyes, and the white eye is a recessive trait. Remember, we're following the F1 generation. This is the gene on the X chromosome, and this is where it's found. And the reason why we're only seeing males with white eyes in this case is because here, when you get this cross, this female here ends up with one version of the wild type, which is dominant, on one type of the mutant. So she's heterozygous here. Okay, so she's got one working version of the gene. The male, however, when he inherits the mutant form of the X, he doesn't also inherit another X to counteract that, unlike the female. And so that's why we're only seeing males with this genetic disorder, in this case, this mutation, because they only have one copy of the X. And we're going to see this later on. We see many genetic disorders that only males suffer from, not because there's anything wrong with males, But because they only have one working version or one version of an X chromosome, and so they're a lot more likely to get a mutant version of an allele and not have the dominant version to counteract that. Females can get away with getting a mutant version of an allele, but it's more likely that they're also going to get a working version in their other X. So they get an extra chance to counteract a mutant allele, but the males don't because they only have one X. And we'll talk about that in a little bit more as well. Okay, so the gene for eye color is located on the X chromosome. And so here we're going to write the gene here. This is the X chromosome. This is the wild type. Remember, W stands for the first mutation that was found in eye color, which is white, and the plus represents that it's the wild type. So this is an allele causing red eyes, an allele causing red eyes. This is an allele causing white eyes. All right, and it's found on the X chromosome. Okay, and this is why we're going to get this situation here. All right. So we're often going to see genes that are found on the X chromosome or the Y chromosome, the sex chromosomes, are going to have some interesting patterns that are going to follow sexes. So we're going to see differences between males and females, basically. Right, one thing we need to just talk about a little bit is what exactly sex is. This is especially difficult for us because we have the word sex and we have the word gender. Like, what do these mean? So in biology, all we're talking about when we talk about the word sex is what, like, gender are you biologically in terms of your chromosomes. We're not talking about, like, what gender do you identify as. That's not what we're talking about. So in biology, your biological sex is determined by your sex chromosomes and what you have. Okay, so it's going to differ from gender, where gender is how you identify, and sex is like what actual chromosomes you have. And this is the case in females. We have X's and Y's. Males are XY, females are XX. Now here's some cool chromosomes. This is Homo sapiens, this is you, and this is a male, so one X and one Y. And you'll see here, this is a homologous region. Homologous obviously means the same, so these are the same genes here. Differential means that this purple bit here is different to this red bit here of the X. And this is a plant. Not quite sure what plant it is, but it's a plant. And they've got something different going on. They've got a larger larger Y than an X. So it's different between different organisms. So here on the X chromosome, you have about 1,100 genes are on here, and these genes don't necessarily cause like sex differences or anything. It's just they happen to be on the X chromosome. But on the Y chromosome, you've got 78 genes. However, you've got some duplicates as well. And then half of those genes are going to be expressed in the testes. So it's actually going to be the Y chromosome that is going to determine whether you're going to be male or female. The X chromosome doesn't really do much. It's basically, do you have a Y? If you do, well, then you're going to be a male, because the Y is going to like turn on the genes that says start making testosterone, for example. This is a really interesting video. If you want to watch it, it's about different types of sex determination in different organisms. And it's just interesting. It's a TED Talk. It's pretty short. It's just an interesting video if you want a bit of a mental break. And then, of course, evolution of animal genitalia is actually really, really fascinating. You should watch that video. It's really cool. So, like I said, the sex determination in humans is not the only type of sex determination. For example, in birds, they have 76 chromosomes, you can see here, and they've got something called the ZY system. Okay, and here you're going to see the difference here. This is a female, the hen, and this is the male, the cockerel, and it's the female that's going to have the two different chromosomes. The male here is going to have two of the same, so it's like the opposite way around. In insects, they have an XO system, O meaning nothing, so does a sperm carry a sex chromosome or not? That's going to determine whether the offspring is going to be male or female. Bees and ants are really cool. They're basically just kind of clones of each other. The male's going to be haploid. They're kind of just walking sperm, basically. And the female's going to be diploid. So you have interesting things going on. The point I'm trying to make is that humans are not the standard, normal way that sex is determined in organisms. It differs between different organisms, and it differs between whether the male or the female is basically going to determine the sex of the offspring. So when is your sex, your biological sex, actually determined? It's going to be around 2 months old. And like I said, if you've got a Y chromosome, you've got something called a sex determining region of the Y chromosome, and it's needed to basically say you're going to be a male. So if you go down here, you're going to start making some testosterone if you've got a Y chromosome. So what we've just seen in Drosophila is something called sex linkage, and it makes sense that because your sex chromosomes determine your sex, well, if you've got particular genes on those chromosomes, which you do, that those genes are going to follow these patterns of sex determination as well. And the question we're going to ask, which is really interesting, and I need you to know this for the exam, I'm probably going to ask you about it, is which chromosome is more responsible for sex-linked genetic disorders and why? And which sex suffers more frequently from sex-linked disorders and why? So why don't you have a little think about this, maybe pause the video and see if you can come up with some good arguments for the answers to this question, which one you think it is. So in sex linkage, we often see males are going to suffer from these sex-linked genetic disorders more so than females. And the reason for that is because, remember when I was talking about mutant alleles? And a mutant allele is just a broken version of a gene. And so, for example, haemophilia is a really interesting example. It's like blood clotting. It's like the gene that causes you to be able to, your blood to clot, is not working. And so it doesn't mean that females can't get it, but it's a lot more common in males, because if a male gets a broken allele, basically, that says you can't clot your blood, and it's on the X chromosome, well then he doesn't get the chance to be heterozygous like the female does and inherit a working version. So normally in our autosomal chromosomes, where we have typical complete dominance, mutant phenotypes come up less frequently because you need to be homozygous. You need both of those alleles to be broken in order to, see that mutant recessive trait in your phenotype. But for males, if it's X-linked, if the gene is X-linked, well, and they inherit an X with a mutant allele in it, well then they're going to get it because there's no other X, there's a Y instead. And also, yeah, there's a lot more genes on the X chromosome. Remember, there's about 1,100 genes on the X chromosome, but on the Y chromosome there was about 25 proteins, and about half of those are going to basically be expressed in the testes and determine that you're a male. And so there's a lot more genes that can kind of go wrong on the X chromosome that could be mutated, and then males get the raw end of the stick for that because they only get one X. So here's another way of writing alleles. If you're not talking about like wild type, if you're talking about just like dominant and recessive and it's wild type, for example, in humans. So in Drosophila, remember we had that wild type with the plus, but like if you're talking about sex linkage in humans, and I used a really stupid example here of a picture of a fruit fly. Now I'm telling you to write it differently. So pretend this is not a fruit fly. It's a mosquito. Okay, so this is a mosquito, and you're not going to talk about wild type or mutant, you just dominant or recessive, you're going to basically do the same thing that we talked about before. You've got the capital and the lowercase, but you're going to write it as a superscript here to the X, depending on which chromosome it's on. If it was on the Y chromosome, then this would be a Y instead. Okay, so this might be, I don't know, something beginning with N. Whether you've got nodes on your branches or not, that's something to do with plants. Okay, so this might be nodes, no nodes. All right, hopefully that makes sense. Right, so let's just do a few problems because I'd like you to be able to do this. So I want you to be able to write the chromosomes or the symbols. So remember, if it's an X-linked disorder, it's going to be an X, and then you're going to have a superscript, and the superscript will be capital or it will be lowercase, depending on whether that allele is dominant or recessive. So, for example, let's say we're following feather colour in birds, and blue is dominant over green, which is recessive. Now, normally, if it wasn't sex-linked, blue is dominant, green is recessive, it would be capital B for blue, and it would be a little b for green. If it's sex-linked, we're going to have an X if it's on the X chromosome, and then we're going to have the superscript big B for blue and little b for green. So you can use the symbol, I don't know, let's just say A, because A is the first letter of the alphabet, so why not? So it's going to be XA, and you want to say whether it's dominant or recessive. So if a father has an X-linked disorder and it's recessive, and the mother is homozygous dominant unaffected, what is the probability of a son inheriting the disorder and a daughter inheriting the disorder? So work out, remember, so it's an X-linked, so it's on the X chromosome. Dad is XY, and then you have to figure out, does he have the dominant or recessive allele? Okay, it's told you it's recessive. And then the mother is homozygous dominant, unaffected. So do this Punnett square, and you're going to do a Punnett square where you're going to do an X with the little superscript allele and a Y for the father, and then you're going to have X, X for the mother, and the symbol you need to work out what it's going to be. Is it going to be a capital A or a lowercase a? So you can have a go with this. Pause it. Do your best to figure it out. All right. So this is the affected father, and the way you would write this allele here would be X with a little superscript A. The mother is going to be X with a capital A superscript X capital A, because remember she was, if we go back, she was homozygous dominant, so she was unaffected. And they've just shown it with different colours here. And so you can see I think they're trying to make an interesting diagram, but, you know, Punnett square would have been nicer. So this is the male here. Remember, it's X-linked, so the Y doesn't really matter. So to his unaffected son, he's going to give his Y, because the son gets the Y, and the X is going to come from the mother. Okay, they're going to have a carrier daughter, okay, because she's going to get this mutant allele here that causes a disorder, but from the mom she's going to get a working version, so she's going to be fine, but she's going to be a carrier, so she's going to be X little a, X capital A. The same with this daughter here. These are the other gametes here, because remember, it's this X with this X, or this X with this X, and then there's going to be an unaffected son. So the son's going to be fine because he gets a working version of the X from mom. Okay, so we call this X-linked recessive with an affected father. All right, now, if the mother was heterozygous for an X-linked recessive trait, so let's just talk about what that's going to look like and then have a go. X big A, X little a, so she's going to have one orange and one blue. So do that Punnett square, see what you come up with. Okay, so you've got an affected offspring here. Here you don't, you just have carriers, but here you have an affected offspring, and that's because she's a carrier. You might not be able to tell whether she's a carrier or not based on her phenotype, but on her genotype, she's a carrier. So this is what this is going to look like. So it's actually really important. It's the female that drives a lot of genetic disorders, because she can be a carrier. And remember that a son is going to get one of her X's. She doesn't get any. The son doesn't get any X from the father, only from the mother. Right, now if the trait is dominant, if the father is affected or if the mother is affected. So again, you could do this Punnett squares, make sure you write it right. The X is going to have a superscript, capital A or lowercase a, depending on whether the trait is dominant or recessive, and what the genotype of the parents is. Okay, so that's what it's going to look like. X-linked dominant with affected mother. X-linked dominant with an affected father. So you just go through the different scenarios, basically. We'll do one on the next page. Okay, right, so haemophilia is X-linked recessive. A carrier woman has a baby with an unaffected man. So you have enough information there to work out what the genotypes are of the parents. Remember, dad is XY, mum is XX. What's the probability of the couple having a girl who has haemophilia? And what about a boy that has haemophilia? So pause the video, have a go, because I'll probably give you an exam question very similar to this. All right, this is what you should have had. So an unaffected father, he has the capital H, haemophilia, because he's unaffected. The female is a carrier, so she has one working version, one mutant version. And the important thing here is that half of the sons, as in you've got 2 sons, 1 out of every 2 sons is going to have haemophilia. Remember, males are a lot more affected by genetic disorders that are on the sex chromosomes, because the X has a lot more chromosomes and a lot more genes to go wrong, and then the male only needs 1 broken version of the allele and then he's got it. End of story. You don't get this situation where you can have, yeah, she's got a mutant allele, but she's also managed to get the working version, and so she's fine. All right, just one more thing I'm going to talk about in terms of chromosomes, genes on sex chromosomes. So, how many alleles does a female mammal have for each sex-linked gene? Remember, male is XY, female is XX. So there's going to be differences between sex-linked genes, number of versions for males versus females. So if a chromosome is on, if a gene is on the X chromosome rather, so if a gene is on the X chromosome, then the female has 2 X's and the male only has 1 X, and so like, what is going on there? What are we going to do? So there's something called X-inactivation, where just one of the X's, one of the X chromosomes in a female, only in a female because she's got XX, is going to be randomly inactivated. And that's called a bar body. It's this little blobby thing here. Basically, so she starts off with XX, and one of them is going to be randomly inactivated. Remember that, yeah, she's got 2 X's, but she could have different alleles on there. So let's say this is haemophilia. She could have haemophilia or not haemophilia. Or this might be blue fur and pink fur. Wouldn't that be great if we get animals with blue and pink fur? So X-inactivation is basically going to inactivate. You're going to shrink down one of these X's to what's called a bar body. And it's not permanently inactivated, because otherwise then every female would only have 1 X to pass on to the next generation. And we know in meiosis that, well, you pass down, you have 2 versions of every gene, and you can pass either version down. And so it's only temporarily inactivated in cells. Okay. So this is a really cool example of X-inactivation, and this is where you get tortoiseshell bird killers. I mean cats, sorry, forgive me. So here is the embryo, okay, and it's going to undergo mitosis when it grows, and then one of those X's is going to be randomly inactivated, and that's represented by the grey here. Now this cat, this kitten here, inherited one X that causes orange fur and one X that causes black fur. And so some of the cells are going to have the black fur causing gene allele to be inactivated, and the others are going to have the orange to be inactivated. So some skin cells, or whatever the cells are that make fur, hair follicle cells, are going to make orange fur, and some are going to make black fur. And this is how you have multicolored cats. Look at its face, it's probably just eating a bird. Look at it. All right, so this is CC here, this little kitten. Okay, look at it. It's seen a bird out the window and it wants to eat it. So this is CC, and CC is a clone of Rainbow. They were very very original when they named their cats. Why does CC not look like Rainbow? Well, she has the same genes. She's a genetic clone, but remember the X-inactivation, it happens later on after fertilization, and the cells are, it's random which X is inactivated. So even in a clone, that randomness, it still happens. So this is why CC doesn't look like Rainbow.